organizers for asking me to give this talk about immune checkpoint inhibitors in pediatric oncology, since this is a very hot topic. There were initially tremendous hope when pediatric oncologists were able to start giving checkpoint inhibitors to their patients, because a real therapeutic revolution was ongoing in adult oncology. And indeed, the very first case reports or series were quite encouraging, with, for instance, the favorable response to immune checkpoint inhibitors of hypermutant glioblastoma reported by the Sick Kids team, or the successful treatment of refractory anaplastic large cell lymphoma by the Institut Gustave Roussy team, and ultimately by the approval by the FDA of pembrolizumab for patients with high tumor mutational burden. But to understand the future, sometimes you have to look back, and sometimes it's not the things that are the most hype that leads to the biggest revolution, as you can see here. And actually, the very first report of a state-of-the-art trial was quite disappointing. And in this phase 1 clinical trial of ipilimumab in pediatric patients with solid advanced tumor, results were disappointing. 3 patients were included, among them 12 melanomas. The toxicities were as expected and according to adults' publication, but there were no partial response, no complete response. The best we could get was stable disease for up to 10 cycles in tumor types that are not our typical pediatric tumors like melanoma, clear cell sarcoma, and singular sarcoma. So while the tolerance was as expected, there were no meaningful clinical activities in pediatric oncology. The next trial about nivolumab in children and young adults with relapsed or refractory solid tumors and lymphoma was a phase 1, phase 2 trial recruiting patients in 23 centers in the US with a classical volume 6 design. Nivolumab was given at a dose of 3 milligrams per kilo, day 1, day 15. 85 patients were enrolled, 75 were evaluable. 12 were included in the paid one that allowed to establish the recommended dose of 3 milligrams per kilo, and 73 in the part B recruited according to different disease cohorts. And here again, these are the details of the side effects, but toxicity was similar to adults. So there was overall a good safety profile. The most common adverse event was anemia in 50% almost of the patient, and fatigue in a third of the patient. 5 DLTs were observed. And the most striking thing is that pleural effusion was seen in 11 patients. So it's something that we had to keep in mind when treating this patient. When trying to explore the immune infiltrate of the tumor, the authors were able to see that with rare PD-1 expression, lots of macrophage, and internal response, it's overall disappointing, except for patients with Hodgkin lymphoma. There were 10 patients included, and that were evaluable for response. And among these patients, there was 1 complete response, 2 partial response, 5 stable disease. In the non-Hodgkin lymphoma, only 1 complete response, but for all other tumors, no other responses. There were only stable disease, 11 for sarcoma and 5 for neuroblastoma. In the phase 1, phase 2 trial investigating atezolizumab for children and young adults with previously treated solid tumors or non-Hodgkin lymphoma or Hodgkin lymphoma, this was performed in 28 centers, both in Europe and the US. Atezolizumab was given at a dose of 3 milligrams per kilo, day 1, day 21. 115 patients were screened, 90 were enrolled, and actually 87 received the treatments. Here again, you have a table with detailed results. Most common adverse events were, here again, fever and fatigue. Most common serious adverse event was fever. Interestingly, 16 patients presented immune hepatitis, so it's globally similar to what's been reported in adults. The interesting thing here is that although PK was comparable to adults, there was a significant lower exposure in infants, but these are only 2 patients, but these need to be further explored. In terms of activity, it's disappointing here again with a median duration of treatment of 0.8 months, so that's basically 2 cycles. Only 4 partial response were observed here again in 2 Hodgkin lymphoma and 1 non-Hodgkin lymphoma, but also in 1 rhabdoid tumors, not confirming all the other rhabdoid tumors that were included. And there were 9 stable disease, 4 neuroblastoma, 2 non-Hodgkin disease, 2 Ewing sarcoma, and 1 Ewing's tumor. And interestingly here, the expression of PD-L1 did not impact of the problem or the likelihood of response or tumor control. And lastly, pembrolizumab was investigated in a phase 1, phase 2 trial in pediatric patients with advanced melanoma or PD-L1 positive advanced or relapsed refractory solid tumor or lymphoma. Over in 30 institutions, 11 countries, pembrolizumab was given at a dose of 2 milligrams per kilo at day 1 and day 21. The important thing here in this trial is that patient was selected on PD-1 expression on the tumor. So we may not choose the same biomarker now, but it's a biomarker-gated trial. 863 patients were screened for PD-L1. 286 patients were eligible, and at the end, 155 patients actually received treatment. So here again, tolerance was good, mainly grade 1, grade 2 effects, although 97 of the patients experienced adverse events. Half of them were related to treatment, mostly anemia and fatigue. There were 8% of grade 3 to 5 adverse events and 80% of immune-related adverse events. Interestingly, here again, in 3 patients, there were grade 5 pleural effusion. So the safety profile is good, similar to other anti-PD-1, PD-L1 agent in pediatric oncology. In terms of response, as you can see here, most of the tumor of the patient did not respond. The interesting thing here is that among the 15 Hodgkin lymphoma patients, 9 responded, 9 did not relapse during all the treatment period and follow-up. So the overall survival is 100%. But this is clearly a big interest in using PD-1, PD-L1 for patients with refractory or relapsing Hodgkin lymphoma. And for all the other tumor types, subtypes, no complete response, 6% of partial response in this kind of tumor. So here again, adult tumors like mesothelioma, adenocarcinoma, lymphoepithelial carcinoma, and tumor reduction that was observed occasionally in all these tumor types. So from all these phase 1, phase 2, and PD-1, PD-L1 trial, we can conclude that children are not little adults, so we have to think differently. And so why is it that different? The first point is that in pediatric oncology, we have cold tumors with a very rare immune infiltrates. That can be modified by metabolic reprogramming, epigenetic reprogramming. That's what we're going to have a look after. The second point is that pediatric oncology tumor have a low tumoral mutational burdens compared to the adult counterpart. So basically less mutation, less neoantigen, so it's harder for the immune system to recognize the cancer cells. We may also have other specificities of the immune system in children, maybe NKs, maybe even microbiome. So there's room here for increasing our knowledge and fill the gap to come up with more specific and rational combination. Because that's the way to move forward to try to overcome the intrinsic resistance to immune checkpoint inhibitors. We go for combo. So in this figure, you can see all the pathway targets, drugs that can be used to modify the immune landscape of a given tumor. And what we have to do here is try to impact on all those potential targets that are not in the white part, because this is to increase the activity of immune checkpoint inhibitors in hot tumors. So there's actually the number of combination trials in immunotherapy and oncology is rocketing. And in this figure, I want you to look at the panel A that gives you the number and types of trials ongoing in adults and children, combining NTP1 and other type of treatment. And the other key thing is in panel D, you see that most of the trial aim at turning cold tumors into old tumors. So we cannot copy and paste other strategy, but in some cases it's still worth it. So this is an example of a copy and paste from adult strategies that very likely will not work in children because you combine NCPD1 and CTLA4 in brain tumors. So PK results have been presented at SPNO last year, and hopefully we should have the final results soon. And these are other examples of combination that can be used in pediatric oncology. So the first example is addition of CTLA4, but different molecule from different pharma companies. So in trying to work nicely in hot tumors, very likely won't work in a cold tumor. PARP inhibitor to increase DNA damage, so to increase neoantigens and the chance of the tumor to be recognized by the immune system. Or you could use other immunomodulators like pegylated interleukin-2. We know that this agent can increase lymphocytes and NK cells at the tumor level, so you have a richer and more favorable tumor microenvironment. Or you could use MEK inhibitors, because MEK inhibitors have been shown to be able to increase the specific tumoral lymphocyte at the tumor and decrease the apoptosis or their death. Another example that we developed in France is the combination with metronomic chemotherapy. So in arm 1, by using capecitabine, we want to deplete MDSC. In arm 2, by combining vinblastine and cyclophosphamide, we want to maturate dendritic cells and Treg depletion. And in arm 3, we combine arm 1 and arm 2 to obtain a multiple immune restoration. And we alternate low-dose cyclophosphamide with low-dose capecitabine and combine it with vinblastine and nivolumab. So the phase 1 is completed. We have 2 partial response in patients with high-grade glioma and RTRT among 10 patients. So it's pretty encouraging. And arm C has been chosen for the randomized phase 2 part of the trial that we compare arm 3 versus arm 3 plus nivolumab. And the last possibilities of combination that is being tested in France and Europe in this ITCC trial, it's the combination of lirilumab and nivolumab. Lirilumab is an anti-KIR agent. It targets the KIR3DL2 receptor, so that strategy allows to combine the targeting of adaptive and innate immunity, and you actually unblind NK by facilitating NK activation. And we know from preclinical and clinical data that NK is important for controlling rhabdomyosarcoma, neuroblastoma, glioma, ovarian sarcoma. So the trial is ongoing. So overall, the use of immune checkpoint inhibitors in children, there was lots of hope, and there are currently lots of frustration and deception. Nevertheless, we have some success in hypermutated tumors, Hodgkin lymphoma, non-Hodgkin lymphoma, and some of our patients that have adult tumors like renal cell carcinoma, or mesothelioma, or choroidoma. The good thing is that tolerance is good. We can use the same dose as in adults. PK are the same, so it's also easier to set quick phase 1 trial requiring a less number of patients. And we were in a first area, I guess, testing single agent treatment, and now we're moving to combo. We now have to find what is the right combo for the right tumor. And there are many, many possibilities to be tested, so we have to be smart and come up with strong rationale. And there are many unanswered questions, such as how about long-term toxicities? Are anti-PD-1, PD-1 the good starting block for immunotherapy in children? we have to be careful with picking younger children, and we have to work and understand better the specificities of our kids' immune system to come up with strong, rational combination. So I would like to thank you for your attention and